I
Hi,
like the
Lisa.
background, Miss Nandi. Hi.
How are you?
Good. I'm lucky you over there at the beach, girl. I see you over there. Who told you to go on vacation?
Yeah, I am in Florida now.
Oh my goodness, we won't have a teacher Wednesday.
You will have, I will teach you from Florida.
Oh my goodness.
So do you like my screen backdrop?
That's nice. It's very nice.
Yeah.
All right.
Who joined? Michael. How are you? I can't hear you. Turn on your audio.
Can you hear me now?
Yeah, I can hear you now.
Sorry.
Yeah. How are you?
I'm great. I'm very, very tired.
Tired? Yeah, me too.
I got out of my shifts at the hospital and it's fun, but man, sometimes you got a bunch of stuff that happens. It just keeps you drained.
Yeah, I know. I understand. Yeah. So how is it going?
Inside the hospital, it's good. It's busy because every person we see, we got to automatically assume the worst. But it's going great. Like I don't know. I have a lot of fun doing it. It's definitely keeps you on the edge of your toes, and I don't know. I love this field. So, what was cool is I had to uh, take some stuff to a lab, and so I got to see this class in action, which
Uh-huh. Yep.
because they were sitting there and they were like, "Yeah, you go over there, you test for everything over here," and to see like an actual compound microscope and them going, "Yeah, we're trying to figure this out," just baffled me.
Good. So now are you comfortable with microscope? The
Well,
focusing and all?
yes. The only part that I ever had the issue with is with the oculars, like initially, but other than that, it's fine.
Oh, that it would be fine because throughout the semester anyway, we are from next week onwards we are going to start gram stain,
Hmm.
and every day you will use microscope. You should be fine. You'll be fine, I'm
Oh
sure.
no, I'm confident in it for sure.
Yeah.
It was just really cool to see this class in action.
Oh, good to hear. Yeah, Lisa,
Yes,
Lisa,
Miss
did
Nandi.
you hear?
Yeah, I heard it. I was thinking, oh, that must be nice. That's wonderful.
Yeah.
It was intense walking in and everyone's in a lab coat and you're going, "Oh my God, I am not sophisticated enough to be in this room."
No. Fine. Yeah, so Lisa,
Yes.
don't be scared.
I'm not scared, Miss Nandi. It's just,
Mhm.
I think I'm more nervous about remembering things from Labster for the final. Um,
Um.
that because I'm not really learning much from that.
Okay. So you think too much. Don't be worried about that final lab final. I am here. We will work together for the labster and the midterm everything. So don't worry.
Okay, Miss Nandi, I trust you, girl.
Remember, watch that video so that way whenever we're using the tubes, we use the correct finger on the thing.
Yeah, that too.
Okay, so what's the time? Let me see how many
It's
people.
time to start.
Time to start? Yep.
Yes, ma'am.
Let me start. So first, minimize.
Going on mute.
So we already started chapter 7, and okay, I think I need to start from proteins, right? Last week I worked up to this.
Yes.
Yeah. So let me see. I think somebody joined. Okay. Okay. So you can see my screen, right?
Yes.
Good. Okay, Lisa,
Yes.
I have a question for you.
Oh, yes, ma'am.
Are you ready?
I don't know, but here you go.
Okay. So, uh, one, It's a pearl necklace.
Uh-huh.
Okay. So if I consider it as a polymer, then what do you say one individual pearl?
You said what?
One individual pearl or bead.
Uh-oh.
Who can tell me?
It's a monomer.
Monum.
Oh, okay, yeah.
Is it Tate? Who said? Who gave me the answer?
Rachel,
Rachel. Oh, good job, Rachel.
thank you.
How are you?
Good. How are you?
Good. Okay, yeah. So that's the review from last week. You know, as I told that all the polymers they are made up of subunit monomer, right? And remember, for carbohydrate, glucose was the monomer, right? For fat or lipids, it was the fatty acids and the glycerol. Now, do you remember all this?
Got it. Yeah, got it now.
Okay, good. Okay, so proteins and other biomolecules, okay, we are going to work on this. They are very large, so they are also macromolecules, like a carbohydrate, like fat and lipids. Proteins, they do lots of so many different things inside the biological system. They acting as hormone, enzyme. They work for the cellular structure, cell processes. They are the signaling molecules. They work as antibodies. So proteins actually involved in nearly every dynamic function in your body. And they are very diverse. Okay. Just give me a second. Banhi has joined. Okay. So proteins, they involved in almost all the cellular process that occurs inside a biological system. Very diverse. Now, all these proteins, they are actually made up of subunit, you can say, amino acid. As I said, the proteins, they're very diverse with tens of thousands of different proteins, okay? So many different types of proteins are there. I told you that they are the hormones, they are the enzyme, they are the antibody. So you can understand that the diversity of the protein or the variety of protein inside now our body. Now the structure, as I told, that protein, they are made up of the subunits, it's called amino acids. And all these amino acids, they are linked, or you can say bonded, with special type of covalent bond. And it has special name, which is called peptide bond. Okay, so basically, so this is one amino acid, then amino acids number 2, amino acids number 3. So all these amino acids, they are actually linked with a bond, covalent bond, which is called peptide bond. Okay. So for the protein, these amino acids, they are the monomer. Okay. So 20 different amino acids are there. So very interesting fact is that, as I told that amino acids, they are the subunits or the monomer of protein. Inside our body, already 20 types of amino acids exist inside our body. And these proteins, they are built from a selection of 20 types of amino acids. Okay. As a result, there are inside our body, actually, there are a great variety of probable protein sequences. Okay. So how many protein sequences could be there inside our body? So if we want to work on, let me get a whiteboard. Okay, so let's say this is amino acids 1, amino acids 2, amino acids 3. Let's say inside the body, there are 10 amino acids, okay? And this is the amino acids number 10, okay? And in between we have peptide bonds. All these are attached with, sorry, just a minute. Okay, date has joined. Okay, so let's say this is a protein and this protein has 10 amino acids, okay? 10 amino acids, and this is number 1, this is number 2, and this is number 10. And in between we have peptide bond. Okay, now as I told that already inside our body 20 types of amino acids exist, okay, female. So if we work on the amino acid 1, so possibilities are there that 20 different types of amino acids could be there. So basically for the amino acids 1, 20 possibilities exist. Do you understand? Could you please turn on your audio? I cannot hear.
Yes.
Okay.
Yes, ma'am.
So
Yes, ma'am.
exactly same way. So 20 possibilities are there for amino acid 2. 20 possibilities are there for amino acid 3, and all the way, if we go to the 10th one, so 20 possibilities are there. So if we do the math, then we can see the possibilities are almost 20 to the power 10, and that I think it will give you almost 13 possibilities. Okay, so you can understand the variety of protein and why there is so much variety. Okay, so now these proteins, let me go back to,
I want to be wherever that is at. Your background was you on a beach.
yeah, do you like that?
That was cool.
She on vacation. That's where she is. She
Yeah.
done dipped.
I told you I am at Florida. I just took lab today morning and now I am at Florida.
You
Okay.
dipped out.
Yeah. Okay. So let me change. Okay. So you got the point, the variety, so many different types, and why there are so many different types of protein sequences inside our body. So now the amino acids. Okay, so what's the next question we can talk about, like what's the differences between all these amino acids, 20 types of amino acids. So what's the difference between one type of amino acids and other type of amino acids? Okay, so to work on that, we really need to work on first the structure of the amino acids. So this is the structure of a amino acid. So amino acids, it is comprised of, there is a central carbon. Okay, this is called central carbon. NH2, this group is called amino group. Okay, this one, C double bond OH, COOH, that's the carboxylic acid group. Okay, I hope you remember all the functional group. There is a hydrogen attached with the central carbon. Okay, and there is one more functional group which is called R group. So these are the component of amino acid. One central carbon, one hydrogen that is attached with the central carbon, one amino group, and another carboxylic acid group, plus one R group. R group, R side chain. Okay, so all these together, they build one amino acids. Now, I asked you a question that what's the differences between all these amino acids? So what happens? The R group actually changes from one amino acid to other. Okay, so you can see over here the leucine. Okay, for leucine, the R group is actually CH2, CH, CH3, CH3. So together, this is one functional group, and I hope you remember the definition of functional group. Last week we worked on. So functional group means all the molecules, all the compounds, they together, they work as a single unit, right? And they determine the characteristics or the chemical character of that component. Okay, so over here, all these together, they are one functional group, which is a R group for amino acids. Okay, but this is another amino acid which is serine. For serine, that functional group or R group, it is made up of only CH2 and OH. For aspartic acid, another amino acids, you can see that R group is built up with CH2, C double bond O, and OH. So basically, all these amino acids, this part from here to here, they are common. Look, that central carbon, okay, attached with the hydrogen, it has amino group, NH2, and carboxylic acid group for all of them. Only differences is this R group. This R group is different from one amino acid to other, and that's how they are different from each other. Okay, and the characteristic features also different from one amino acid to other. Are you clear?
Yes, ma'am.
Yes.
Yes.
Yep.
Should I move?
Yeah.
Okay, so order of the amino acids in a protein determines the 3D shape and the function of the protein. So basically, at this moment, you all know that there are 20 different amino acids, okay? And I told you the sequence, there could be so many varieties of, are there inside a cell. Now, not only the amino acid, the presence of amino acids and the number of amino acids and the structure and the arrangement all together, the chemical features or chemical nature of protein actually changes. Now, in chemistry, what happens, all the molecule, chemical compound, their function actually depends on the structure. It's in chemistry for each and every molecule and compound. Okay, and the structure determines the function. Exactly same way for the protein, the arrangement of all those amino acids, basically the structure, you can say the shape of protein determines the function of the protein. So if we lose the structure, then we are going to lose the function of the protein. Okay, and it's very easy to lose that structure if you can heat little bit of changes in the pH, temperature. In changes in the temperature actually change that protein structure. Okay, and change in protein structure means changes in the function of the protein. Protein, if it is high heat, high acidic environment, then protein cannot work. Okay, and that is called, that characteristics features we actually represented by this word. It's called denatured. When we change the structure of the protein by increasing or decreasing the pH concentration or temperature, salt concentration, protein cannot work anymore. And then we can tell that the protein got denatured. Okay. Somebody has joined. No. Okay. Okay. So protein can have 4 levels of structure. So now you know that structure of protein actually determines the function. Inside our body, the protein can have 4 levels of structure, or 4 different shapes and structure. They can exist in 4 different shape and structure. These are called primary structure, secondary structure, tertiary, and quaternary structure. So let me get a whiteboard so to be easy to draw. Okay, so protein structure. At this moment, you learned that protein, they are made up of amino acid, and now we know the structure of amino acids. Okay, so if I just give me a second. Okay, so amino acid and protein structure. So if we want to draw the protein structure, it's actually, so let's say this is the amino acids 1. Okay, then we have carboxylic acid group, and again amino group, and this is amino acid 1, this is amino acid 2. So it goes like this way. Amino acid 3, and let's say, so over here, this is the carboxylic acid group. So this is the structure of protein. Okay, and in this protein we have 3 amino acids, and this is this part, that actually forms the peptide bond. Okay, so inside the peptide bond, you can see that carbon is attached with the oxygen and nitrogen is attached with the hydrogen. So as I told, that there are 4 different level or structure of protein. Okay, so primary structure, so primary, secondary, tertiary, and quaternary. So these are the 4 structure of protein, okay? And this is the basic idea, or you can say basic configuration of protein, how the amino acids remain bonded together with the peptide bond. And every time, you know, the structure of protein, when we write, we start from the N terminal of the first amino acid, and actually we go towards the C terminal of the last amino acids. So that's the way you can draw or you can write the protein structure. Okay, so now there are 4 levels of protein structure. So the first one, the primary structure, it's a linear sequence of amino acids. Okay, you can see over here that you can see this is the primary structure of protein. Okay, so over here, let's say these are the amino acids. So over here they have bond. It's a linear sequence. Inside the peptide bond, we have carbon, oxygen attached with a nitrogen, hydrogen. So this structure, the primary structure, is very simple, pretty simple over here. Now, for secondary structure, what happened? If we see the structure of peptide bond over here in this structure, so carbon is attached with oxygen, and over here nitrogen is attached with the hydrogen. So inside this peptide bond, both the oxygen and hydrogen, they can form hydrogen bond. They are capable of making or forming hydrogen bond. Okay, so that's the peptide bond, and this oxygen and this hydrogen, they together are capable of making hydrogen bond. So this is the linear structure of protein, and over here we can say, look, multiple peptide bonds are there. So we can say this is a polypeptide chain. Okay. Now, so inside a polypeptide chain, we have hydrogen and oxygen, and that oxygen and hydrogen, they are capable of making hydrogen bond. So what happens when there is a large polypeptide chain, and over here we have all those peptide bonds, this hydrogen and oxygen, because they can make hydrogen bond, that formation of hydrogen bond with this oxygen and hydrogen, that actually fold the primary structure, primary linear structure of protein. Okay, and that folding can bring into 2 different structure. One is spiral shaped, you can say it is called alpha helix, and another one is folded structure. Okay, and this is called beta sheet. So because of the formation of hydrogen bond inside the peptide bond, and that actually helps to hold that linear structure of the protein, and that can gives the secondary structure of protein. And the secondary structure, 2 different shapes are there. Because of that formation of the hydrogen bond, 2 different shapes can be achieved. One is the coil or spiral shaped, that's the alpha helix. Okay, and another one is folded, that's called the beta sheet. Now inside the alpha helix, what happens, that formation of hydrogen bond, sorry. Okay, so that formation of hydrogen bond actually gives that shape over here, and that hydrogen bond occurs inside just 1 polypeptide chain. okay, in case of alpha helix. Now, in case of beta helix, what happened, that formation of hydrogen bond can occur inside just one polypeptide chain, or maybe between, so let's say over here, oxygen and hydrogen. So it can occur between two different polypeptide chain. Okay, the result is same, the folding of the polypeptide chain and different shape. So when that formation of hydrogen bond occurs only inside one polypeptide chain, and it gives a spiral shape to the linear structure of protein, that is the alpha helix structure. And these hydrogen bonds actually very weak bond, very poor bond, but over here together all the hydrogen bond, they can bring the stability inside this spiral structure, inside this alpha helix structure. So the difference between alpha helix and beta sheet, in case of alpha helix, that hydrogen bond form only inside one of the polypeptide chain. Beta sheet, either there is only one polypeptide chain, or there could be two polypeptide chain, and attraction occurs between the hydrogen and oxygen of two polypeptide chain, and that creates the formation of hydrogen bond, and as a result that folded structure of protein can occur. So that's the difference between alpha helix and beta helix. Now the tertiary, third level of protein structure, what happened over here, protein actually that secondary structure of protein actually folded multiple times, and that gives a 3D shape of the protein. Okay, so basically we can say the arrangement of protein over here, arrangement of protein, or you can say that amino acids in 3D space, that actually gives us that tertiary structure. So over here, as I told, it could be like this, much more folding than the secondary structure. So we started, so from primary to tertiary, we started with just one linear polypeptide chain, okay? And we got the spiral shaped alpha helix, then folded beta sheet, and then the 3D shape, that's a tertiary structure. Now for protein, protein is not, they cannot function until they get this three-dimensional structure. That's the very characteristic features of protein. Okay, to be functional, protein needs to get that 3D structure. Now the next one, fourth level, that's the quaternary structure. Now over here, that's actually the interaction between multiple tertiary structure. Okay, so what happened over here? So let's say this is one tertiary structure. Okay, then it can interact with another one. It can interact with another one. It can so that's how they make the quaternary structure. Okay, so when only two tertiary structure there, they form the dimer. When there are three tertiary structure, they make the trimer. So that's the way the interaction goes on, and they give this quaternary structure of protein. So we have very nice example inside our body, that hemoglobin inside our blood. They have 4 tertiary structures. So basically 4 tertiary protein structure, they interact with each other and they gives the structure of hemoglobin. Okay, so the hemoglobin that actually carry oxygen, that transport oxygen, very important protein inside our blood. That's the example of quaternary structure. So that's very popular and you know that name, but that's the reason I talked about hemoglobin. But most of the protein inside our body, functional protein, I mean, they live in quaternary form. Okay, immediately after protein synthesis, you can find out the linear structure, the primary structure or the secondary structure, but most of the functional protein inside our body, they remain in quaternary structure, quaternary form. Okay, so these are the 4 level of protein, or 4 different structure of proteins. They exist inside our body. Let me go back to the PowerPoint. Okay. I already talked about this. Okay, so I forgot one thing to talk about is, so secondary structure, I told that secondary structure occurs because of the formation of the hydrogen bond, right? Tertiary structure, what happened, yes, Lisa?
I said we don't see the PowerPoint.
You don't see the PowerPoint?
No.
Okay, let me go back. But I'm sharing that screen.
There it is.
Now you see?
Yes.
Okay, so for the secondary structure, hydrogen bond was responsible, right? Inside the peptide bond over here, that interaction between amino group, carboxylic group, hydrogen and oxygen, they make the hydrogen bond, and that is responsible for this alpha helix and beta pleated sheet structure. Now, for the tertiary structure, that hydrogen bond forms, that is one reason, but there are a few other interactions occurs. Okay, another one is called disulfide bond or disulfide bridge. Okay, so what happens, so let's say this is one primary structure of protein, and this is the another one, or maybe the part of the protein, you can say. Okay, so over here, the portion of the primary structure, interaction between two sulfur molecule that create disulfide bond or disulfide bridge. There are another interaction can occur, which is called hydrophobic interaction. So in between, you can see so in between the chain, the polypeptide chain, that hydrophobic interaction can be there. So that's another reason of this folding. Then the ionic bond, formation of ionic bond. So because of all these different type of interactions, so hydrogen bond, disulfide bridges a disulfide bond, then hydrophobic interaction, then number 4 ionic bond. Because of all these interaction, we get that tertiary structure. Okay, so I talked about everything. Do you have any question based on the protein structure? Primary, secondary, tertiary, and quaternary. Did you get it? Are you
Yes,
clear?
ma'am.
Yes.
Do you have any question?
No, ma'am.
So for the test, make sure that you know that primary structure, that's the linear sequence of amino acids, okay? And they're linked with the peptide bond. Then over here, hydrogen bond is responsible. Then tertiary structure, remember that hydrogen bond, then disulfide bridges are there, then hydrophobic interactions are there. It could be the ionic bond. These are the reasons. For quaternary structure, these are the interactions between multiple tertiary structure. Pretty easy to remember. Okay, so you don't have any question? Okay, so we are done this chapter. Do you have any more question from this chapter? No? Okay.
No, ma'am.
No, ma'am.
So let me start next chapter, okay? Okay, so we are going to work on chapter 8 now. Do you see my screen now? Chapter 8?
I do.
Okay, so this chapter, chapter 8, talks about microbial metabolism. Means this chapter is actually going to explain how microbes, they get nutrients, they get energy to survive and to reproduce. Okay, this chapter is mostly about that, how microbes get their nutrients and energy to survive, to sustain life. Now, this metabolism, you will see that in this chapter, we'll work on so many different strategies that microbes actually do. There are a variety of different strategies microbes do, and that's how. So basically, different microbes they do metabolism different ways, completely different process, and that's how we can differentiate between the different species of microbes. Okay, so this metabolism is very important characteristics to identify or to differentiate between the microbes, and this is very important characteristics of microbes. Like, you know, like how we decide that these microbes they are very harmful, or these microbes they are very helpful. We actually decide based on them, on their metabolic processes, or the product of the metabolism, based on the product of the metabolism. Now, the product of the metabolism is called metabolites. Okay, so different organisms, different microbes, they produce different metabolic products, or different end products that sometimes harm us or sometimes help us. Like, you know, that nitrogen fixing bacteria in leguminous plant, that nitrogen fixing bacteria, what it does, what they do, they actually convert nitrogen into ammonia. Okay, and that is very, very helpful for the plant, because plant, they cannot utilize this inorganic nitrogen. Okay, but that ammonia is very important for the plant to growth. Those bacteria, they are called nitrogen fixing bacteria. They reside inside the roots of the leguminous plant. And they convert that nitrogen, inorganic nitrogen, and into ammonia. That's ammonia is the the result of one type of metabolism. Okay, and that is very helpful for us. On the other hand, in the mining process, there is one group of bacteria. They are called sulfide oxidizing bacteria. What they do, the sulfide oxidizing bacteria, during that mining process, their metabolic end products is, you can say, the one of the byproduct is sulfuric acid. Okay, that sulfuric acid kills so many different plants and animals because of the mining, that presence of sulfuric acids inside that mining drainage. Okay, so that's very risky or harmful for us. Like now the coronavirus. Okay, so COVID-19 virus. Just last week, one of my cousin sister, I just called them in India. So she told that she was very sick because troponin. Okay, so one of the chemicals inside her body, that was very high. It got up to 372. Actual limit of troponin is only 100. Okay, so that's the normal limit of troponin. But my sister, certain, one day that troponin level was too high. She was suffering from breathing problem. Okay, so she told me that she couldn't breathe and she feels like very weak, she will fall down like that. Then they took her to hospital or nursing home, and then they did all different type of blood tests. They did ECG, echocardiogram for the heart, everything, and then finally it came out that heart troponin level was really high. Now that troponin is it actually represents, when the troponin level is high, it represents that there was heart attack. Okay, so to find out the heart attack, if there is any attack on the heart, usually we test that, we check the troponin level. Okay, so for heart, at the beginning, immediately after doing the blood test, they found out that her troponin level was high, so they thought she got heart attack. So they did that ECG, echocardiogram, EKG, they did, and they found out that nothing had happened. The heart was pretty normal. It's not for the heart. Then finally, that cardiologist, he told it's a COVID attack. Okay, the virus, like what happens? Somehow they entered inside her body, and then another one was a little high on her blood, that was neutrophils. Okay, so that cardiologist explained that COVID-19 entered inside her body. Okay, that neutrophils, that how actually that neutrophil present that viral antigen to the other white blood cells. That's the reason the neutrophil was high in number. So her immune system was pretty good. So immune system could destroy all the viruses, but little portion of viral toxin was remain inside the blood, and that toxin, okay, actually attacked that heart muscle, and as a result troponin level was high. Okay, so that viral toxin, it's a metabolite, it's a product of viral metabolism. Okay, so these are few example I just gave you to understand that why that metabolism is important and why these metabolic products are very important for us. Okay, so the metabolism in biochemistry. Let me pull a whiteboard so I can write down. Okay, so in biochemistry, the word metabolism means the series of chemical reaction that occurs inside a cell. So if I write it down, the series of chemical reaction that occur inside the cell, okay, and why it happens to sustain life. Okay, so altogether, all these chemical reaction, or you can say the total of all these chemical reaction together is called metabolism. Now the question arises that why do we need all this chemical reaction, or why do they occur? So the answer is pretty easy: to sustain life. Okay, then the next question, how did this happen, or who takes part in these chemical reactions? Okay, so we have all the four biomolecules. You can say the carbohydrate, protein, lipids, and the nucleic acids. Nucleic acids means DNA, RNA. Okay, so all these four biomolecules, they do so many different types of life-sustaining chemical reactions to promote life, and that's the metabolism. Okay, or we can say that brings the metabolism. So for all these series of chemical reaction, which we say that metabolism, who are responsible? This carbohydrate, protein, lipids, and the nucleic acid, we can say that. So what they do, they do series of chemical reactions or series of life-sustaining activities, you can say, to promote life, and that's the metabolism. Okay, so now that carbohydrate, protein, lipids, nucleic acid, so how do they come inside our body, or from where do we get all these? Okay, so one source, when we eat food. So we eat food, and that actually our body get all this carbohydrate, protein, lipids from the food, right? Okay, so what happens? Let's say the food, when you eat your food, let's say it's a chicken. Okay, so you have to chew, you have to make it small. If I give you a big chicken breast, you cannot put the whole chicken breast inside your mouth, right? So you have to cut it small, then you can take small pieces, and then you start chewing, so to make it much more smaller. So why actually you are chewing? Because to make it small, because you really do not have enough space to put the whole chicken inside your mouth. Exactly same way, inside our cell, cell needs, through enter inside cell or to go outside of the cell, you understand that that plasma membrane is there. So it required only small particles, very minute particles can move in or move out from the cell. Okay, so because our cell, they are so minute, what happens when we eat food? All these food particles needs to chew or needs to go through the process of digestion. Okay, that actually breaks down the large food particles into the smaller one. Okay, so that's a process, digestion, that breaks down the large food particles, and that gives this, that breaks down this carbohydrate, protein, lipids, and nucleic acids, large macromolecules, into the simpler form. So what happens after digestion? That carbohydrate, it breaks down into glucose. That's the monomer of carbohydrate, right? So that's the simplest form of the carbohydrate, and digestion do that. Then protein, it breaks down into amino acids. For the lipids, it's breakdowns into fatty acids. Nucleic acids, they breaks down into nucleotides. These are the monomer of all this. Now, why do we need to break down all these large macromolecules into the simpler one? Why? Because our cell, to assimilate inside our body, because of our growth and reproduction, all those things, cell cannot actually have all those. They cannot accept all those. These are pretty large molecules. So that's one reason of digestion, to break it down into simpler form. But there is another reason, because all these things actually can convert into again these large particles when our body needs. So basically over here we are breaking down the large particles, and then we are reconstructing our large particles from the simpler forms. So these two things, they're basically inside our body, there is a serious balancing goes on between this process, breaking down the large particles into the simpler one, and the reconstructing the large particles from the small particles. So these two actually couples with each other, and as I told, there is a serious balancing goes on between these two processes. Okay, so now these two processes, so when the process actually that breaks down the large particles into the simpler one, okay, that process is called catabolism. Okay, now from simpler molecules, these are like Lego pieces, you can say. These are the Lego pieces. So reconstructing the large particles from all these Lego pieces, that's called the anabolism. So all this catabolism or anabolism, like when the digestion is going on, that's so many different type of chemical reaction goes on. And to rebuild the larger molecules, like during the anabolism, so many different chemical reactions that goes on. Okay, so together, this anabolism and catabolism together, they are called the metabolism. Okay, now that's the simple definition of metabolism, because over here that was very broad definition, series of chemical reaction. So that's why I tried to break it down to make it simple. Okay, now I told you that our cells are so small, and through the plasma membrane, that large particles cannot move, and that's one reason we breaks down the larger particles into the smaller one. That's true. That is one reason. That another reason is there to produce energy. Okay, so when we break down these large particles into some smaller one, we also produce ATP. ATP, that's the energy currency in our cell. So whatever the work you do that requires energy, reproduction, cell growth, development, for all these works, we must have supply of energy. That energy actually comes down when we break down these large particles into the simpler one that produce ATP. Now, ATP, what happens? ATP breaks down into ADP. So ADP is adenosine triphosphate, means 3 phosphate groups are there. It breaks down into ADP. That gives us adenosine diphosphate plus inorganic phosphate. Okay, so basically it breaks down one phosphate bond, and that phosphate bond is high energy bond, and that releases a huge amount of energy, and that's how we can do work. Now, ATP is very essential, okay? So that ADP must convert into ATP whenever body needs, okay? So basically to continue this circle or continue the production of ATP, ADP must join with the inorganic phosphate, and that's how they they continuously working on to build up ATP. So ATP breaks down into ADP, and then ADP again converts into ATP when our body needs. So basically based on our need. So this thing also goes on. That's also part of our metabolism, and that's the one purpose of metabolism. Okay, to produce energy so that you can do work. So in this chapter, we are going to study this catabolism. We are going to study the anabolism. We are going to study the ATP, ADP. Now, few more definitions. Okay, so I told you that breaking down the large particles into simpler one, that's a catabolism, and reconstructions of large particles from the smaller ones, that's a anabolism. Now, these two, this catabolism and anabolism, they couple each other and couple together. And the other thing, these two reactions are controlled by enzymes. That's another protein. Okay. We are going to, in this chapter, we are going to learn a little bit about the enzyme. Now, this process, when we break down large compounds into simpler one, okay, and we produce energy and we produce oxygen, that's the cellular respiration. So that's another purpose of metabolism. So in this chapter, we are going to work on the cellular respiration. Cellular respiration means inside our cell that that glucose is 6H12O6 that actually breaks down, that becomes oxidized and produce oxygen, carbon dioxide and ATP. Okay, so that's the cellular respiration we will work on in this chapter. So that's the concept of metabolism. So this catabolism and anabolism, they goes together. Okay, they are dependent on each other. During one process, we produce ATP, and during other process, we breaks down ATP to release some energy based on body's need. So all these things together is called the metabolism. So let me go back. Okay. So the metabolism total of all chemical reactions occurring within a living cell in order to sustain life is called the metabolism. Function: assembles smaller molecules into large macromolecules needed for the cell, degrades macromolecules into smaller molecules, like when we break down carbohydrate into glucose. Okay, energy is conserved in the form of ATP. So all these occurs during metabolism. As I told, that two process, anabolism and catabolism. Catabolism, the word that catabolism, make sure you understand. Pretty easy way I used to remember any catastrophic event. So any catastrophic event, so lots of destructive things happens, right? So catabolism is the destruction or degradation of large molecules into the simpler one. Okay, so that's the catabolism. So over here during catabolism, what happens? Uh, like let's say C6H12O6, when it breaks down into carbon dioxide, water. So that's the catabolism. Over here, we had 6 carbon, 12 hydrogen, and 6 oxygen, but that breaks down into oxidized into carbon dioxide. Pretty simpler one. And that's an example of catabolism. In our life, the cellular respiration, that's the example of catabolism. Anabolism means biosynthesis. We are forming large molecules, okay? Like when plant does photosynthesis. So what plant does, what they do, they grab carbon dioxide from the air, then they grab water. With the presence of sunlight, they make C6H12O6, completely opposite of this one. So over here, from very simple, just one carbon molecule, they make 6 carbon compound. Okay, so that's the example of anabolism. Now, over here in this reaction, you can see that it has to be, this reaction needs sunlight. Sunlight means that provides energy. Okay, so these anabolic reactions, they must have supply of energy. To continue the reaction, anabolic reaction, we must have the supply of energy. On the other hand, during catabolism, it generates energy. Like over here, when we break down C6, one molecule of glucose, we produce carbon dioxide, water, and at the same time, we produce 34 to 36 ATP. So that's the production of energy. Okay, clear the differences between anabolism and catabolism? And I hope you understood that how they are interdependent on each other, not only the anabolism, catabolism, and how the ATP and ADP is related with this metabolic process. Okay, so just a second. So now we are going to work on the energy. Okay, so what is energy? Energy, capacity to do work. Whatever you do, that requires energy. So when you are sitting, Okay, just sitting, not doing anything, that also requires energy, because you can understand that your head, it's so heavy, right? And that heavy head, it has, weight. So you have to hold this head straight against the gravity. Okay, that requires energy. So when you are lying down, when you are sleeping, that requires energy. So capacity to do work that actually allow you to do something, like when you are sitting and holding this head upright against the gravity, because your body is producing or providing energy. Energy in salt. So how we utilize or how we produce energy, we have two different types of chemical reactions: exergonic reactions and endergonic reaction. So the word exergonic, okay, pretty easy to remember. Exergonic means exit. That's how I used to remember. Okay, so exit. So in this reaction, energy actually releases. Okay, so basically, when the reactants, let's say these are the reactants, A plus B, they start the chemical reaction and they produce the product which is called AB. At the end of the reaction, this exergonic reaction, they release some amount of energy. Versus in case of endergonic reaction, endo means inside. So over here, to start the reaction, we must have supply of energy. So in this case, A plus B just simply cannot produce the product AB. We must have supply of energy at the beginning of the chemical reaction. Then if we have supply of energy, then A plus B together, they can make AB. So for in exergonic reaction, the reaction release energy over here. Endergonic reaction, it needs energy to start the reaction. Okay, so that's the difference between exergonic and endergonic. And as I told, that pretty easy to remember if you remember exit. So that's exergonic. That's the only thing you have to remember, nothing else. Okay, so now what's happened over here? Let's say you have $40. Okay, you have $40 in your pocket. You went to mall or somewhere. Okay, so you bought $30 of clothes and all those things. Then you can have that $10 extra that you can use for, let's say you want to eat ice cream. So you have $40, so that's the reason you have that $10 extra to eat ice cream. Okay, so over here, how do we get that energy from? Energy, we cannot create energy. So energy, we cannot create and we cannot destroy energy. One type of energy can be converted into another type of energy. So it cannot just fall down from the tree, that energy. So from somewhere it has to come. So this energy was stored within these reactants. Okay. So these reactants, they had huge amount of energy stored, and that's the reason they can release extra energy, just like your $10 ice cream. Okay. In this case, what happens? We must have supply of energy at the beginning. So basically, you have $40 over here you didn't use, and that $40 remains over here. Okay, I'm coming to that point later. So over here, what happens for the reactants? You know, as I told, that energy, all these two reactants, they stored energy. They had high amount of energy, and extra energy they can release. So basically, in this reaction, the reactants, they carry high amount of energy than the products. Okay, so that's another characteristics of the exergonic reaction. So reactants, they have much more energy than the products. But over here, in case of endergonic reaction, we at the beginning itself, we supply more energy, okay, to do the reaction. So basically, the reactants, they don't have energy to start the chemical reaction. So that's why we are providing this energy, okay? And all this energy is going to use to produce the product. So in this case, the product actually stored lots of energy, more energy than the reactants. Because one thing, remember that—
Miss Nandi,
Yes.
I am so sorry.
No, tell me.
We still see the whiteboard.
Oh, really?
Say it again. Yes, I've been sitting here going, this isn't making sense.
Oh my God! Why did you tell me?
Yeah, we haven't been able to see anything since you started talking about energy. I've just been writing everything down.
Do you see it now?
Yes, ma'am.
Oh, okay. I'm going over again. So you understood the metabolism, right?
Yes, ma'am.
Yes.
Okay. So this slide you didn't see at all?
No.
Oh, okay. Okay, so I'm starting to work. So energy means, you know, the capacity to do work. So anything you do that requires energy, right? Inside a biological system, inside our living cells, there are two different types of reactions goes on. One is exergonic reaction, another one is endergonic reaction. In case of exergonic reaction, what happens? So let's say this is the chemical reactions, okay? So A and B, these two are the reactants. These two together, they produce, this is the product, AB. So these two reactants together, they produce AB. That's the product, okay? In this reaction, what happens that A and B has enough energy so that after producing the product, after finishing the chemical reaction, they have extra energy that they can release. So in this reaction, exergonic reaction, at the end, this reaction releases energy. Whereas endergonic reaction, to start the reaction at the beginning, to start the reaction, there must be supply of energy. Okay, if there is no supply of energy, this chemical reaction cannot go on. So at the beginning, to start the reaction, we must have supply of energy. So that's the differences between exergonic and endergonic. So exergonic, that energy releases at the end of the reaction. Endergonic, at the beginning, to start the reaction, we have to have supply of energy. Now, in case of exergonic reaction, what happens? So we release energy, right? Remember I told that we cannot create energy, we cannot destroy energy. Okay, so this energy, from where do we get this energy? It has to be here. okay, inside the reactants, right? That's why after doing all this work, we have extra energy, and that we can release, right? Because we cannot create energy, right? So what happens in this case, in the exergonic reaction, the reactants, they actually store a huge amount of energy, okay, more energy than the product. And that's why after completing the work, they can release extra energy. Okay, so that's the exergonic reaction. So the reactants are much more stronger, means they carry more energy, you can say, okay, than the product. Product carry very little amount of energy. So all this energy is needed to complete, to create, to make the product, and then still they have extra money on their hand. So that's the release in the form of energy. So that's the exergonic reaction. Now, endergonic reaction, what happens that A and B, the reactants, they don't have enough energy to start the reaction. So we have to supply extra energy to start the reaction. So let's say we had 40 energy. Okay, now to produce the product AB, you know, it requires 30 energy. To produce, to do the work and all those, and then 10 energy get lost. So over here, in this case, AB, the product, it cannot actually store any energy, because 30 energy, we started with 40. So 30, we had to use to produce AB, and 10 energy. Any chemical reaction, we actually lose a little bit of energy in the environment, so in the form of heat or some way. So we really do not have any money on hand at this moment. So in this case, in endergonic reaction, A and B, they really did not have any money, so they are much poor than the AB. Okay, so that's the difference between exergonic and endergonic. So in case of exergonic reactants, they are rich in with high energy than the product. Okay, over here they are very poor, so the products products are very rich than the reactants, because to do the work we had to supply that energy. So 30 energy already we used to produce AB, so it has 30, versus over here they did not have anything, so that's the reason we had to supply extra energy. Okay, so that's the endergonic reaction. So are you clear the differences between exergonic and endergonic?
Yes, ma'am.
Okay, so the example, the exergonic reaction, the respiration, cellular respiration. Okay, for cellular respiration, what we do, we break down glucose, right, and we produce energy. So that's the example of exergonic reaction, versus the photosynthesis. The plant does that photosynthesis. What happens at the beginning? We supply that sunlight in the form of energy, right? So that's the endergonic reaction. And I hope you got the relationships or the status of the reactant and products in both of these reactions. Requirements for carbon. So based on, so for all organisms actually, two things are very important, okay? Carbon and energy. And based on the carbon requirements, or you can say the source of carbon and the source of energy, we actually can classify all the organisms, okay? So over here, so requirements for carbon. So based on the source of carbon, we have two groups of organisms. We have autotrophs and heterotrophs. Okay, autotrophs use inorganic carbon. I hope you remember from the chapter 2, I think we worked on the organic and inorganic. Remember, when there is carbon hydrogen bond, that's the organic. Okay, when there is oxygen hydrogen bonds, that gives us organic. When there is, you know, the only hydrogen is attached with carbon and separately with oxygen, it's that hydrogen oxygen is not attached, or carbon oxygen hydrogen is not attached, that gives us inorganic. So over here, like carbon dioxide, what happens in the environment? We have enough supply, huge amount of carbon dioxide in the environment, but we don't have hydrogen, we don't have that carbon hydrogen bond over here. So carbon dioxide is actually one inorganic compound. Okay, so the autotrophs, they can utilize this inorganic carbon. So from the environment, they can grab that inorganic carbon. And that's there, that inorganic carbon is their sole or principal carbon source. Just like plants, they can, during photosynthesis, they can grab that inorganic carbon. Versus the heterotrophs. Heterotrophs, they need organic molecules, okay? Usually from other organisms. Just like us, we are heterotrophs. We cannot consume that inorganic carbon dioxide. We cannot utilize. It depends on plant. What plant does? Plant absorb and get that carbon dioxide, then converts that carbon dioxide into glucose, and we can get that glucose, means that carbon in the form of glucose from the plants. Okay, so we are the heterotrophs. So the prefix auto means self, who can make, and trophs mean nutrition, so who can make their own nutrition. Basically the plants, they can make their own food. The other way to describe the autotrophs. Heterotrophs means who, the prefix hetero means who, you know, from others. So we collect our food, means over here we are mentioning the carbon. Basically we are talking about carbon source. So we get that carbon in the organic, as organic form from other organism, from the plant. So we are the heterotrophs. So the organism who can utilize inorganic carbon, they are the autotrophs. The organism who cannot utilize inorganic carbon and they need the organic molecules as carbon source, they are the heterotrophs. So plants, they are the autotrophs. We human being, we are the example of heterotrophs. Now based on the energy, okay, electron source. Based on energy source, we have two groups, phototrophs and chemotrophs. So basically there are two sources of energy. So we can get energy from the light, basically the sunlight. So that's the phototrophs. Photo means light. Or we can get energy from different organic or inorganic molecules. Okay, so basically the chemical compounds. So there are two energy sources for light and the chemical compounds. So the organism who can utilize that light, that they are the phototrophs. The organism who get their energy by breaking down the chemical compounds, they are the chemotrophs. Okay, now you can understand that when we are talking about the chemical compound, so of course there are two different types of chemical compounds, right? Organic and inorganic. So based on that one group of organisms are there who only can utilize organic compounds. Other group of organisms are there who can utilize inorganic compounds. So based on that, we have lithotrophs and organotrophs. So organotrophs means they can get their energy from the organic compounds. Okay, just like us. We can break down glucose by the process of cellular respiration, and we can make ATP. There are so many different bacteria that they are protozoa who are also organotrophs. Lithotrophs, they use inorganic compounds. Microbes, you will see that purple sulfur bacteria, they are actually lithotrophs. They break down inorganic compounds to make their electron, the energy. So they are the lithotrophs. So now for test, I can give you, like, let's say, based on the carbon source and the energy source, if I give you, let's say, auto lithotrophs, means the organism, they use inorganic carbon source, and their lithotrophs means their electrons comes from the inorganic compounds, inorganic chemical compounds. So both of their energy and carbon basically comes from the inorganic source. And here for the energy, they don't use the light, but instead they use inorganic chemical compounds. Okay, so did you get it? Are you clear this part?
Yes, ma'am.
Yes.
So if I give you this type of question, like, let's say, if I give you, let's say, photo, if I give you photo heterotrophs, if I give you photo heterotrophs, and if I ask you to tell me the carbon and energy source, can you do that? Who can tell me? Photo heterotrophs. Who can tell me? So photo, of course, we are talking about the energy, right? So we know the phototrophs, they use light. Okay, so energy source is light. Now heterotrophs means we are talking about the carbon, and we know that heterotrophs are those who use organic carbon source. So for this group of organisms, they use light for their energy, and they use organic molecules for as their carbon source. Good?
Yes, ma'am.
Okay, so if I give any word, if I make any word like this, from here, you can do that, right?
Yes, ma'am.
You can find out the carbon source and energy source. So when you are going to study this slide, make sure don't try to just memorize, okay? Try to understand the concept. So carbons are just two types, auto and hetero. So plant, they can consume inorganic carbon dioxide, so they are the autotrophs. We cannot consume inorganic compounds. Everything inside our body, it's pretty easy to remember. Everything, living soul, human beings, we need organic things. So we are the heterotrophs. Then energy, either it comes from the light or it can comes from the chemical. Okay, so if it is chemical, we have two types, right? Inorganic and organic. If it is organic chemicals, then they are the organotrophs. If it is inorganic, then they are the lithotrophs. So if I give you autolithotrophs, that means their electron or energy source is inorganic, and their carbon source is also inorganic. But if I give you lithoheterotrophs, that means litho means inorganic, so energy source is inorganic compounds, and heterotrophs means for carbon, they depend on the organic molecules. So you can expect one question from this slide. Do you have time? Oh no.
7:02.
Yeah. Okay, so let's keep up to that slide. I will start next day from this slide. Okay. Do you have any question?
Oh man.
No? Okay, so I will see you on Wednesday.
Hey, everybody.
Okay.
Right.
Bye. Good night.